bottom, and so there are small canals that go all through that body. Oh, here's a coral as we're speaking. And so they basically rely on food-sized particles that are smaller than a cell. All the food that they consume has to be taken within the cell, whereas a coral has a mouth, an equivalent of a stomach, and so maybe there aren't enough large food particles like zooplankton in this area. So that's a guess.
Great, thank you, Scott.
Because the current's pushing sideways?
Yeah. I think the current's increasing.
So we just saw another Exocoelactus anemone.
As we've gone upslope.
And there's also been a couple of crustaceans for this dive. A couple of squat lobsters here and there, and also a couple of crinoids living commensally on some of these sponges. We've only seen one type of fish so far, Synephobranchus eel. But apart from those things, not very much else. Sort of a lot of the same around here. We can also see a couple dead stalks of sponges lying around on the seafloor, or also still attached to some of these boulders.
So, Diva, I'm keeping my heading into the current.
Okay.
Just makes it easier.
I'm happy to report our Internet 2 feed seems to be back.
Great. Sorry, what was that, Josh?
So I'm keeping my heading into where I think the current's coming from.
Uh-huh.
To help me keep my heading. Otherwise, it wants to spin me sideways.
Okay.
Just so, like, you're seeing me, like, saddle forward and sideways.
Yep.
I'm really sort of going more to my port.
Okay, sure.
Yep.
Can we have a snap zoom on this anemone, please?
All right, come on in, video. Hold that.
Yep, so this is another one of the Exocoelactus anemones. I think this is our third one for the dive. As well as just behind that we can see a crustacean of some kind. If we tilt up, will we see the crustacean just behind? Yeah, so just there. So that looks like a little squat lobster. That at first glance does appear to be on a rock, but no, it's actually living on a dead sponge, which may actually have some manganese coating on it.
We need another move. Same direction, okay?
Sure.
Yeah. Bridge nav. Yeah, same move again, 30 meters, bearing 185, 0.2 knots. Thank you. Thank you, Bridge.
Snap zoom. Okay.
It's amazing how many dead remnants of dead sponges there are left.
There's a lot here.
Yeah. Interesting.
This dead sponge.
Lots of dead sponge skeletons, hardly any corals, but then still some quite large sponges, as well as lots of predatory sponges. And if you're ashore, we apologize for our poor feeds at the minute, poor streams. We have been having some communication issues with the internet leaving the ship today. Hopefully they'll be rectified soon, and dive today and for the next 3 dives of this expedition, the last 3 dives, will be without problem.
Do a quick zoom. Is that a sponge?
It does look like the base of a sponge. Yeah.
Right.
Dead sponge base.
You can come out.
Looked like a sea star from a distance.
Yeah, that's what I was thinking.
And Deb, the geologist, has just snuck in from her lunch break and said that she'd like me to mention the lovely pillow basalts that we're seeing here. And again, a lot of these dead sponge stalks and dead sponge skeletons are still hosting many of the animals living on them. Comatulid crinoids, barnacles, and other small species. Oh, are these 2 crinoids on top of each other? No, one is a brisingid and one is a crinoid on this. So it's so interesting. You can see here 2, right in focus right now, 2 dead sponge stalks that have very extensive communities of animals living on them. So, okay, so here we've got 2 comatulid crinoids, the brown and the yellow, several munidopsid squat lobsters, which are the white crustaceans, some hydroids, which are the sort of plant-like animals living on the actual skeleton itself, sort of wispy looking, and then this beautiful pink animal, which does look very, very similar here to a crinoid, is actually a sea star or an asteroid known as a brisingid. And all of these animals are filter feeders, and what they do is that they try to get themselves as far off of the sea floor and up into the water column, away from that benthic boundary layer where the water meets the sea floor and there's the highest amount of friction. And so by getting further up into the water column, there's more currents rushing by with more food particles, and they can sustain themselves much more easily.
I'm going to come out just a touch.
So Chris, since you're the sponge guy online right now, could you tell us why we see
Video's clear.
many commensal organisms colonizing dead sponge stalks, but not very many on live sponges? We do see some on live sponges, but just not in the huge abundances that we see on dead sponges.
Let's come in on this guy as we go by.
Sure.
Well, I don't know if the sponges have some kind of defense.
Co-pilot, do you hear me?
I can't imagine what that would be,
I think I can
but
balance
it's
it here.
possible they have some kind of defense. But the other thing, too, is when the sponge dies,
Is this
a
deck A?
lot
No,
of
nav.
stuff starts to grow
Copy.
on it, as you can see with this example here.
I mean,
And that
We're
stuff
monitoring
might
the,
provide food for a number of
hydraulic
these things
comp.
that colonize it, so they wait
Currently
until
at
the sponge has
16%.
got
copy that.
bacteria
If it
and
drops any more,
hydroids
we
and
observe it to
perhaps
drop from that.
forams
We're
and
gonna
everything
work with
else
that.
before they jump on
Copy.
and then start feasting.
I've been watching. I
Great.
got the,
Thank
I
you,
got that
Chris.
Throw
passed down.
something else
Okay.
out there just as a possibility, and that is, as you mentioned a few dives ago, One of the things that
Okay, take a bit
sponge
more
biologists or certain sponge biologists, natural product chemists are
this move's
interested
complete.
in is the secondary metabolic compounds that sponges produce.
Looks to me like
see
there
if they
isn't
might be
really a
useful in
pronounced local high.
any kind of drugs.
Sort of a ridge.
We've been
And
following a
those may
ridge.
be there
Bearing
as
about
some
185
sort of
most recently.
defensive compounds, so maybe some of the chemicals that are
Yeah,
associated
PDA.
with the tissue. And we describe
That still
sponges
seems about
as
right.
generating a current so they can pull water in through the body. And so the more stuff that's sitting on them, the more
it's
that
right.
stuff
I mean,
will
it's kinda
interfere with the
flat
flow.
up here.
So perhaps some of the chemicals,
Okay,
the
we'll do
compounds, the
another 185.
biochemicals in the sponge
Bridge
are
nav.
somehow noxious or toxic, not necessarily toxic,
Yep,
but maybe
another move,
noxious to the
please.
organisms.
30 meters, bearing
one.
185, 0.2 knots. Same as last time.
Yeah, sure. That's
So you
that's
notice
as
my
good a
heading
guess
is off
as
Thank you.
to the starboard.
anything.
Say again, pilot?
This
You
is
notice my
Patty
heading is
at
off
the
to the starboard.
ECC Hawaii, and it's very interesting that there's this
And-
quite
oh, you're
different
off
look
the
to the
track.
character of the seafloor with a
Current
lot
line.
of
I've turned my
fine
heading to
sediment
starboard.
or
The reason I'm doing that is I have a current.
fractured
Okay.
fractured
In that direction.
material. I don't
That's
know,
pushed my
to
tether way
the
out.
right-hand side
And
of the
it was
screen,
making it very
in between
difficult
what
to
looks
keep my
like
heading.
remnants
Okay.
of pillows.
Resumes. So you can maintain your heading at the direction we're moving,
Okay.
which is what
185.
On
And
the
I'm
right-hand
just gonna
side of the
saddle
screen,
around back and
pivoting
forth,
to the right.
adjusting my heading for current, not necessarily where I'm going.
Now it looks like the current's coming from your left.
Oh, I'm moving to my left.
No, but even when you were standing still, it was...
Yeah, it might have shifted. I'm not sure.
Ooh. Okay, so we've got this beautiful Caribbean shrimp just swimming into view right now.
Come on in.
He's moving too quickly. So this is likely from one of two families, Paneidae or Aristidae. And you can see they've got these modified pleopods that they use to swim very gracefully, as well as extremely long antennae, which it uses to pick up vibrations and other things in the water column.
There's a lot of dead sponge stalks.
Yeah, there are, aren't there, Pilot? And a lot of them are covered by manganese, which says that they must have been here for some time.
I mean, just
Oh,
as far
yeah.
as the eye can see, just it's like a forest of them.
Interesting. Oh, we've got another lepidisis perhaps here. Our second or third
Still in
coral
video?
for this dive, as well as what looks like a couple cladorhizid around it, or nope, another small coral. Brilliant. Okay, so this is the highest abundance of corals that we've seen all dive. There are two in one place.
Coming in partial.
These do look like
Yeah,
they
very
may be
nice.
bamboo corals, and you use the name Lepidisis, which is the genus name for bamboo corals that are unbranched. But it's very interesting to see the small one and the large one here together, because one of the things that my students and I have found with the genetics is that these unbranched forms of bamboo corals are present in many different genera. They're actually
Now,
quite
what's
diverse.
the bearing of
And
the
one of
ship
the
now?
things that can really fool us
185.
is they may
185.
grow as a whip when they're young, but then put out branches later. And so especially for smaller unbranching bamboo corals, it's extremely difficult from video to give it a name. It may be totally fooling us. And especially, think about these living hundreds of years. It may take them a long time before they develop a branch.
And a good few more of these really large polyopogon sponges coming into view now.
So I heard the exciting news from the dry lab a few minutes ago that we actually saw two corals at once. It's pretty exciting.
Nah, they just take away habitat and substrate where sponges could have grown.
It's funny that you mentioned.
Right on top of the pilot.
It's funny that you both mentioned that because
So, pilot, I was just
we
told
were
by Nav.
just having a chat outside about how
Pilot, I
even
was just
though
told by
this
Nav
may
that,
have not been the density
if I draw the shop anymore to recover.
of organisms that you had
told that?
hoped for, at least Chris, you were getting to see a lot of
Okay,
sponges.
that's just what Nav told me.
Excuse me.
Who's also.
I'm in sponge heaven right now.
Jim told you that?
And
Yeah.
as usual,
Despite
Patty
the
and I
lack
are in.
of
Oh,
forms, I always find it interesting.
So we'll just pay attention to it.
On dives like this,
Yeah,
to
I've got
note
it to
that
plot
there
up
are,
on
is a
my
large absence
display.
of corals. And
Oh,
you
good.
asked earlier,
Copy, co-pilot.
why aren't we seeing them? And we don't,
But instead of that, we're just moving along at about 185, doing pretty steady 30-meter moves. Last one was about 5 minutes ago.
Oxygen, temperatures, current
Yes.
flow, the amount of food, and all of that sort
It's
of
been
stuff.
that
The
for
more
the
of these
past
sorts of observations we make,
maybe
the more
200 meters.
Hopefully we'll get to understand what is it that's controlling these distributions.
Okay.
And hydraulics, it looks like, have stayed in 16
Thanks,
for the
Scott.
past half hour.
All right, watch lead. We had gotten a little bit behind on that last zoom, it looked like, so
Okay.
I got us caught up here, and if there's anything in view immediately here, we can look around, otherwise I can slowly continue.
if you would slowly continue, that would be great.
Okay.
We are trying to just keep up a nice steady pace as we move along this ridge.
Yep. Sounds good. I'll just do that then.
All right, pilot, would you
Can
be
you guys
comfortable with
give us an
calling
idea
it a
of
move?
what
30 meters?
the
Just to keep everything
direction
moving?
the current is
I don't see
going?
anything
I can certainly ask
Hold on.
in a moment when they're ready to go.
What? I'm ready. What do you need?
We wanted to know if there was a current down here, and if so, what direction it's coming from.
Oh, okay. I'll lift up for a little current test while I talk to Nav here.
Okay, great. Thank you.
It's coming from my starboard, it seems, which is the west.
So coming from the west?
And I'll tell you how fast, or give you an idea. Actually, it's blowing pretty good right now. Just wet thumb in the wind, maybe half a knot.
Okay, thank you. All right,
Yes,
so
pilot. Are you good for 30 meters at 185?
shore-based scientists, the current is coming from the west, and it might be
All right,
about
I'm gonna call
a
it
half
in.
knot.
Yeah. Do you want to do 185 or do you see
So
these
going
two
to the
bumps
west
over
at
here?
half knot, is that what you said?
I think it was coming from the west
Do you
at a
see
half
those
knot.
two little bumps on the sonar on Seirios off?
Okay, come from the west,
Yeah, would
thanks.
you want to center up on this?
Yeah, well,
What's going on? I'm sinking really quick for some reason.
oh, wrong direction.
There we go.
I think that one bright spot might be D2.
Yeah, there's that one bright spot, but you see these two things here?
Yeah.
So as we move along,
turn port
in
towards
addition
that.
to seeing pillow basalts with some fairly fine-grained black sediment
Head
between
that
them,
way.
which is
I don't
actually
really have very good
different color. We're
heading
not really seeing the
control
same
right now. I
sediment
think
that
I
we were
might-
seeing earlier
You
in
got a
our
lot of
dive.
heading up there?
Yeah.
You're not tugging
We
me, I'm
are
okay.
continuing to move
All
through
right,
this
well,
area
it's
that's
current.
mostly
Maybe
sponges.
current's dragging the tether.
Yeah.
We
Just pulling
also
you.
see a
Looks
number
like
of dead
it's
sponge stalks
So,
So
yeah,
that's
do
at,
what you can.
things on them.
Judge the offset.
I'm not sure what things are
So
yet.
do you want to do maybe 170?
Is there
It doesn't
anything else in the
perfectly
field
line
of
up,
view
but
right now,
starts
shore-based
to move you
scientists?
closer, or do you want to do a direct shot at those two bumps?
170 is fine.
All right,
Yeah.
170 at 30 meters, calling it in.
Let's just keep it going here.
Is there anything in particular
Bridge, this is Nav.
that you'd like to zoom on? We've looked at a lot of
All
sponges
right, can we get a
today.
ship move of 30 meters at 170 degrees at 0.2 knots?
I'm not
Good
seeing
copy.
anything right here, Deb.
Okay, thanks.
All right, I'm getting stretched out here, so
Thanks, Bridge.
I'm going to slow down a touch.
I think today's been a great exercise in different types
You're
of
lined
sponge
up on
morphology.
160.
It's been enjoyable to get to see
So I
what
need to kind of
what
start moving
shapes,
further.
which I assume often might be
170
related to different families or species, but it's been very
I'm
interesting thus
adjusting
far.
to 170 pilot.
Thanks. Yeah, there's a big sponge.
I think I can hold that.
Lots of
Whoa,
sponges.
is that a sponge?
Looks kind of like one to me.
Can we go check that one out?
We will. It is up current from me, so I got to push hard
Take
to
your
get
time.
there, but I will get there. Just takes a little bit.
All right, sponge
Yeah, that
experts.
ain't no sipping sponge up there, although I hope Lipton Tea doesn't try to sue me for infringement of copyright.
understand you're having trouble there, co-pilot. Do what you can, and I'll make do.
So I believe we've seen this sponge earlier. Chris, can you tell us what kind of sponge this might be?
I'm going to land with all this current, so just give me 1 second.
So this is in the family Pheronematidae, and we think it's in the genus Poliopogon. A couple features about these sponges: they're massive. They're some of the biggest sponges that exist on the planet.
Wow.
Secondly, the members of this family all attach to the substrate via a very fibrous type of attachment mechanism there, and that's referred to as a lophophytus attachment, where their spicules are all sort of loose and thread-like, and they're not all glued together like other sponges that form a more solid base. So attachment to the substrate is a real key to identifying certain types of sponges. And then third, you see this sort of mesh in between tissue here.
Yep,
and
that's
these
what we're
are
looking
generally
at.
referred to as sieve plates that are covering the oscula or the openings in the sponge. And perhaps that's a protective function. It's not really clear. But this particular group has fairly extensive sieve plates over the front end of it, and that's another clue.
There's 170.
Great.
Is this a demo sponge or a glass sponge?
Yeah, this is a glass sponge, so all of the spicules are going to be siliceous, made of silica.
Move just up or to the right. Either, yeah, that's great. Thank you.
Thank you, Chris. And we've been doing some zoom-ins as we did. I think we saw a squat lobster.
Seconds here, and,
and we're looking now actually exactly at that mesh feature that you were talking about that's covering the oscula.
Okay. We'll do 5.
That's right. That's what's called a sieve plate.
2, 1.
And Chris, if you wouldn't mind just throwing the name of this into the event log, that would be fantastic.
All right, I'm going to push hard to get caught up with you here.
And we're just going to keep moving along. So pilots, what we're most excited in is if by any chance you see coral, but just because we haven't seen a lot today.
All right, if I see a coral, I'll stop.
Okay.
For now, I'm just trying to
zoom along
and get back on track. There's another one of those same sponges to the bottom right there.
Yeah, there are a lot of sponges in view. And that's really what we've been seeing along this entire dive is lots of sponges of several different types.
Do you know what monitor that one is, my fisheye?
And we want to try to make sure we stay sort of towards the top of the ridge.
What?
And we want to follow the biology.
Yeah, I want to change it to the Titan. So that if I can't...
You can change the bucket to the Titan.
Okay, it's monitor 7, the label.
Unless you need the bucket in view.
Well, I thought I was supposed to monitor that.
I'm going to come back towards you just a touch. Oh, you're on 143 now, just so you know.
Yeah.
So I pushed kind of far to the starboard, but that's all right. Current's prevailing towards our port, so I'll get forced along.
And so I'm sure that you might have heard this recently, but we are here at Delcano Seamount. It is a Geo, which is a flat top seamount created when the top was above the surface of the ocean. This is a Cretaceous seamount, somewhere between 65 million and 145 million years old. And that may account for why it has so much manganese crust on these rocks.
Roger.
And what we've been doing is moving along a ridge from north to south, and just characterizing the site for biology. So I am guessing that might be an anemone. Biologists?
This is an anemone of sorts. Look at that current. See that?
Yeah,
see that
now you can really see the current.
Illustrative. Okay to zoom?
Yep.
Anyone want to take a guess as to what this anemone might be? And my understanding is that the way that we identify many of these anemones is by looking at the tips of their tentacles. Wow, that one looks like it's going to be blown away at any moment.
Yeah.
The bearing's 160, right?
The ROV is also getting blown away.
Right now, Copilot, it is 170, but we'll probably adjust maybe to 160 or more for the next move.
However,
looks like
the
you're getting
actual
more
identification
to
of
your
an
port.
anemone is that you collect it, preserve it, and then do histology. It's actually the ultrastructure of the sheets of tissue that stick in or project into where the gut is.
I'm just going to hold here a minute until you get 70 and the tether's out of your face. And then I won't push too far ahead.
So, Chris, I think I just lost... sorry, Scott, I believe I just lost you. But what I heard before I lost you was that it's actually not the tentacles that are the identification. It's that when you take a sample and you do histology, you look at the tissues within the anemone. That actually is what gives you the
maybe
ID.
160 and center up. Those returns are getting
Yep, that
more
sounds
vivid now.
Copy that now.
Can anybody hear me on the telecon right now? I'm getting a lot of feedback, so I just want to check.
Okay, I'm going to lift up here.
Okay, I'm pilot nav 160-ish.
Copy that. This is a really nice high current day. I'm going to get a really good reading today.
Deb, this is Kelly. Just to note, they're going to try dialing into another telecom line to see if that helps.
Yep, just heard that from Roland. Thank you. All right, so we might be having some issues with comms on the ship, but just so you're aware, we are trying to switch lines right now, so hopefully we'll be back with you in just a few minutes. And
it's hard to fight
while I'm by myself, I will talk a little bit more about this really beautiful geology.
I would like to do the next move maybe at 160.
Giant sponges.
It's not directly to those two returns further out, but there's some
It's
stuff
late,
in there.
it's not a little delayed.
Maybe split the difference.
Oh, can you hear again?
Zero's okay with me.
All right, I'll do 30 meters at 160.
I also don't mind if you just want to make a hop right
So, Scott, you were telling us about histology.
How far out is it?
I think I made it to the fact that it was the tissues within that were of interest. Oh, but I can ask about close-ups.
They're about 25,
Pilot,
just
could
below
you
25.
zoom in on the sponge?
Between 20 and 25.
Yep, right in front of us.
Yeah, sure thing. Just slow the bus a little bit here.
Sorry about that.
That's okay.
Yeah, hi, Deb. Before I answer you, I'll just make sure. Can you hear me clearly?
I absolutely can now.
And we'll get settled up here.
Well, that is great. To accurately describe...
Did you say you just want to do a jump right to?
So I want to make a comment here. I think you're seeing a case where a sponge overgrew the rock that it had landed and settled on, and when it got large enough and the current was strong enough, it tipped over on its side.
Really? So is that why we see the spicules on one side only of this sponge?
That just means the spicules aren't in the shadow.
Yes.
So we're zooming in the spicules right now, and there's...
Set me up to say yes, and I didn't even hear what you said.
It was yes anyways, I think. I have no idea what these things are in the spicules.
There's a little, some kind of brittle star or two in there.
All right, so there's definitely a brittle star and maybe another brittle star.
There's a whole ton of them in there.
And then, so I believe what Chris was saying on the telecon was that this sponge probably started out on a rock, it was growing upwards, and then it actually outgrew the rock and fell over. And this is, Chris, is this the same kind of sponge we were just looking at with the mesh over the oscula?
Yeah. I want to do...
just making sure that the...
Oh, sorry. I didn't put the move in yet, but yeah, we're getting returns kind of... We're 160. We have some returns in and out on both sides.
Yes, that's right. So it's going to be a polyalphagon, might be a different species, but you can see those sieve plates over the openings on this one.
Yeah.
And I think the rock, the sponge and Gen rock both fell over. I think that rock there in the front is the one that it rolled with.
That is quite possible. So there is another giant sponge coming into view. Chris, would you like to zoom in on this one
All right,
too?
Pilot, I'll do 30-160, and we'll see what we got.
30 at 160I. I'm off to the port for a second, just in case they want this. We're going to shoot it with the lasers. And
Bridge, this is nav.
Current to make my way back.
Can I get a ship move of 30 meters
Patty.
at 160 degrees?
What's the word on
at
this?
0.2
Can we
knots.
zoom in on it, please?
Yep, briefly. I got to
Yep.
catch up.
Okay.
This current's making things a little difficult.
Good
That's
copy.
great.
Thanks,
I'll let
Bridge.
them know as soon as we're done zooming on this.
I think my half knot was conservative.
Oh, really?
Yeah. Every time I pick up, I get blown around really, really hard.
So here we are at yet another large sponge.
Okay, you can come in, video.
Oh, and it has, is that a shrimp?
There is some commencing. Yeah. Yes, there is a shrimp. Top left. There you go. Come in a little more. I can't quite get the shrimp yet.
You don't know, but I've actually been challenged to correctly identify biology so that I'm attempting not to misidentify everything I see.
Some shrimp guy there. There we go.
And, oh.
This current's picked up considerably since.
So this is yet another different species of Poliopogon. So there's a whole bunch of these things in the same genus that we really don't have names for yet. This is one that we call Poliopogon species B, I think.
Awesome.
Yeah, I'm not going to go forward much. I'm going to turn into the current and drive.
So shore-based scientists and those of you who are following along at home, it turns out that our ROV pilot's estimate of current was actually probably quite conservative, and we're actually being blown around quite a lot. So he's going to need to keep moving a little bit for right now. So even though we can certainly ask for zooms, and I will, I think we need to catch up a little bit right now.
Would you put one of the port or starboard?
Yeah, you can ask for all the zooms. We'll get them. I'll just
on
either
monitor
have to,
7.
I'll just have to judge how long we can spend.
Perfect. Thank you.
Yep. We'll make sure we get everything we need.
I want to see the bucket.
Could change this to the bucket.
So we're just continuing to move along. I mean, this has really been amazing. We've seen so many sponges on this dive, and so little of anything else, really. We've seen a number of these dead sponge stalks where in those little tiny clusters we do see some of the suspension feeders like the crinoids and the Brazingid and the one day I'll get it right, and the hydroids and things like that. But overall, what we've really seen are these individual sponges at a somewhat regular spacing as we move along the seafloor.
So let's bring up this guy here, and I'm going to give you northing and easting speed, and if you type them in your computer there, it'll tell you how fast the current is. So let me push over towards the right. And I'm going to let go of the stick, and we're going to
Patty had mentioned this a little bit before, but we've been going through areas where there's some very specific areas that appear to be more fractured or fissured than others. So we had just been moving through an area that actually wasn't very fissured at all, and this one looks much more fractured.
Which
use the fins.
Fins, got it. Yeah, I'm ready to.
Okay, I'm going to push down a little.
What did I miss?
Definitely just an approximation, I think.
Yeah. Well, it's closer than my approximation, I think.
So for those of you following along,
if we could take a look at that tipped over sponge up ahead if we have a chance and are going that way. It's just another example of a sponge that's knocked over probably by this current.
I will absolutely ask.
It doesn't, current looks like it actually isn't as bad as it once was back in that one spot.
That's all right. We'll see what it gets up to.
So I think, so pilot, if we have a chance to get over towards what's now in the far center right,
yep,
or maybe top right,
grab those.
It's getting really far away though. It's like a fallen over sponge.
Northing 1.4,
I think it's
easting
losing its way.
0.14, 0.38.
Oh, you can really see the manganese crust here too.
All right, so you have
how it's sort of formed almost like a pavement. Yep.
There are 6.
I'll see if I can. It's a little out of range.
Well, that's good to know.
Wow, that's a total speed of 0.45.
I think it's one of those two that's still in the background, probably the one
degrees.
that's now in the center top.
How many knots does it say?
It gives it meters per second.
Okay. There's the scalers.
I don't know about the rest of you, but I'm very
left
excited
on
to
the
hear
wall,
that there are 6
down.
parties in conference right now. Now I have to start counting to see who those parties
All right,
are.
so now I have the thrust at 100% left or 100% right, and the current is holding me steady.
Oh, really?
Yep.
So we've got a ripping current up here. You can see it going by.
When I was eating lunch, I was.
Yeah, we were talking about the current.
Just absolutely being.
Did you see the anemone?
Yeah.
It was like sideways.
Yeah.
So what we're hearing from the RV team right now is that they've got their thrusters are on just about as much as they can go, and they're having a pretty hard time making headway right now.
We're more efficient forward than we are
like panning.
left and right.
Like, yep. Okay.
So I can still keep up going forward and back just fine, but if I put my side to the current,
you're going to get turned.
I can't make up ground.
Okay. Great to know.
That's
and I'm not sure if we said this already, but perhaps this extremely strong current could be why there are so many dead sponge stalks.
And actually, I believe there was also a comment about whether this strong current might lead to turbulent flow that the corals don't necessarily like.
Perhaps.
And the last sponge was knocked over in a different direction than this one, so maybe these are tidal and go back and forth all day. Just as another interesting note.
Yeah, certainly this sponge is knocked over in basically the opposite direction of the old sponge.
Here's another case of an animal that unfortunately settled out on a rock that was a bit too small for this type of current environment. And once it got big enough, I suspect that the current just blew this over, and now he's on his side and may have a bit of trouble from this point on. And I think it was either you, Deb, or Diva just mentioned maybe why there's some dead stalks laying around.
I got a little excited.
But it did so well for so long, Chris. Look how big it is.
It is a very big sponge.
Looks like it just ended.
Okay, you can keep that going.
All right. Do you want to keep moving at?
Yeah, 160.
160.
Yeah.
Copy that.
Yeah, I'm holding that heading.
Seirios can hold it.
You just can't take settling on the correct rock too lightly.
Words of biology wisdom
Never
to live
should
by.
move up 30 meters at 160 degrees
geology
at
wisdom.
0.2 knots.
Settle on the right rock.
I already got carpal
Good copy.
tunnel,
Thanks,
I'll
Rich.
stick to the right all day.
There's a paper that talks about this sort of phenomena that we're seeing with the sponges about, I think it was black corals, some kind of coral on fjords, on hills of fjords up by Vancouver Island.
Yeah.
And when they started to grow on these rocks up at the sill of the fjord, they grow to a certain height, and then the current would knock them over. So they would continue to grow, but kind of in an L shape to resume growing upward, such that they essentially were forming a cupped hand, and then they would grow big enough that the current would catch them and pull them right down off of the sill and below the depth at which they could effectively
Is that a
feed.
coral? Coral whip or a sponge whip?
That is very cool, Scott. It sounds a lot like those trees that kind of will grow out of the side of a cliff and then turn their way to continually grow upward. It sounds like it probably would look the same.
Can we have a quick zoom on the
On the whip?
Yes, please.
Yep, absolutely.
Yeah, but
Did we find a coral?
3/4 of a
I
knot.
know, so this might be our fourth or fifth coral for this dive.
Hold in this current for a second.
And we've got some coral experts that cannot wait to see this.
Lepidisis, maybe.
I can land and get a closer look in a second. I'm just waiting for Seirios to catch up a little bit. Bounce. Getting a tether bounce. Let me land. Come on out wide. Alleviate the tether bounce. Let's not land on that huge.
So, shore-based scientists, we are trying to get a zoom on that coral. We just have to figure out how to get there. What's that other thing in the left-hand corner that's going off the screen right now?
There's a big... the other whip. Yeah,
Another
there's another
coral.
small whip there.
Are you on? You're so quiet.
Oh.
Better? Yes. I think that
I just
actually...
have my quiet voice on today.
I think that may be the first time that I told you you were quiet this entire... It's like entire expedition.
Yeah.
Thanks. So watch lead.
Yes.
We did a more numerical analysis on the current, and our last test we got about 3/4 of a knot.
Oh,
definitely variable.
What direction is it coming from?
It's coming from the west.
Okay, still from the west?
Yeah, right? Is that what you got?
Yeah, coming from 70. Or, I'm sorry, going towards 70, so yeah, from west to east, roughly.
Great.
So.
Now look at that big sponge off to the right, too. Oh. All right, so coral folk, this is for you. Kinda.
Kinda. Sorry, settling.
It's okay. And Jeff, I know you're doing the best you can.
Yeah. Well, there's loose rocks on the bottom too, so you set down and then they roll over a little bit.
That's full.
Just rolling the rocks around on the ground. Awesome.
So Scott thinks this might be from the I2 clade, based on its thin, tall polyps, but it has very long internodes.
And that means its nodes are spaced very far apart, I assume?
Video. I'm ready when you are.
Full wide?
Thanks.
Yep.
What's this little
Deb, so if you look at the structure of that skeleton, you see that it alternates white and black.
Oh.
Let's zoom on that unusual texture of seafloor right in front of the
We've got everybody talking at once right now. Pilots, can you zoom down on the rock in front of you?
This rock here?
Yeah, just in general. I think maybe not on the rock, but maybe on the broken up rocks. I'm not sure exactly what. Patty, could you write in the event log what it is you were trying to look at?
I heard something about the texture.
Well, the texture is the manganese crust. I assume that those are like scars from where there were sponges or corals. Diva, is that true?
Yes, so those would be the dead sponge skeletons.
So it's still spicules that are left
bigger,
there?
But yeah, so now there's just a remnant left of what
we've seen a lot of that. So Patty, what were you interested in seeing as we zoom in on this rock? Maybe not quite that far. Ooh, what are those?
Little baby.
So those look like the same sponges which we collected yesterday. Remember the little ones?
Baby sponges?
That had the white dots.
What I was interested in was an area on the seafloor that was in front of that.
Come back out. Area on the seafloor, what?
I don't know. Patty, could you write it into the event log? We're having a really hard time hearing.
I only have a second longer here. So
and we might have to leave, so if you could type quickly, that would be great.
I'm going to go back to where we were. Here's the coral.
I mean, to me, it looks like we're seeing a manganese crust, like cemented.
All right, fighting to get.
The spot she's talking about just came into view, but.
So here's a patch of something different. Take a quick zoom.
Is it this grayish area?
Yeah, that looks almost like sediment.
Oh, this is not a good time. Oh, no.
Like if that was sediment, I'd say it was bioturbated, but that's clearly not what's happening here.
I would doubt it. Yeah, if we could zoom in on that.
Yeah, that's it.
Yep. That is very interesting.
Is that full zoom?
Not yet.
Can we zoom on that tubed animal? Possibly a Serianthid.
Trying to hijack our geology
well,
moment.
And you'll see the geology more closely.
Longer here. Oh, and that's a tether pole.
I really don't know why that patch is discolored compared to the rest of this manganese crust with sediment on it.
There's a ton of current.
Very interesting.
Below me, maybe a little behind me.
Okay.
So it looks clean now. Tether looks good.
All right, I have to run ahead.
Okay, thank you.
Sorry.
You're off to my starboard.
Thanks. All right, so unfortunately we have to keep moving. We just were looking at a mysterious patch on the seafloor that was very gray compared to the rest of the light brown sedimented manganese crust.
Yeah.
So we're being sort of pushed along by the current.
Off sticks.
Over this area of sort of a manganese-encrusted volcanics. And we are hanging out in sponge land. Dead sponges, live sponges, many sponges.
How do you know you're
sponges
not
with stuff
run
on
into a
them.
boulder on the left?
What's that?
How do you know you're not going to run into a boulder on the left?
Very carefully. I was just here.
Oh, okay.
I had to backtrack to get the gray. Someone's got a radio on in the control room. I can hear myself.
That's funny. I was just about to ask you why we're hearing an echo of you.
Yeah. Can you make that go away? Is it gone now?
No.
Tell. I don't know where that's coming from, but
we here in the control room are very lucky. We get to hear the ROV pilot on multiple channels right now, I think.
Make sure that check your right knob video and make sure you push down so it lights up green all the way to the right there on the top. If you're on the headset, make sure that's green. Well, it sounds like it's behind me.
Oh, sounds like it got fixed.
There it goes. Great.
Oh, good.
Sorry, it was actually.
All right, so right now we have do we have about an hour left in our dive, Diva?
Yes, we do. Exactly an hour.
We have about an hour left in our dive.
Straight for move?
185. We've been doing 160.
We are at a depth of 1,840 meters right now.
Maybe more.
We are
closer
continuing to move towards the south as we move along this ridge.
It's kind of flattened out here.
We've definitely gotten into an area that has more sediment, less manganese crust. Not sediment's not the right word. Sorry, more broken up rock. And are those more dead sponge stalks, or are they something different?
No, so they do look like dead sponge stalks.
Okay. Yep. And a sponge. I'm shocked. We found another sponge.
I'd suggest we go 180.
Okay. 180 is good for me. I'll sidle port for it.
Bridge nav.
I'm basically facing 180 right now.
And we're facing just about south right now.
Please, 30 meters, bearing 180, 0.2 knots.
We're just moving along, trying to characterize this area of the Delcano Seamount.
Working the lats out.
Yeah.
Good today.
Yeah, we're gonna.
Good copy. Thank you.
I was excited to see
what's
what
that in the center right there
tonight.
All of these just look like dead sponge stalks.
Even
and and a whip coral.
Oh, in the middle?
Oh, there. Right. Sorry. Again, I was looking at the wrong screen. That looks like a Brazingid on a dead sponge stalk, perhaps.
Okay.
Perhaps.
The next move we called in was 180 pilot.
Copy.
So a couple of crinoids, comatulate crinoids around here. Some more dead sponges.
Just a couple minutes ago.
Of various different types. And you can just see how strong the current is in this area from the arms of these crinoids.
It's got long legs to hold on.
Those are called Seirios.
Long Seirios.
And it does. Very, very good observation. I'm surprised it's able just to hold on to that rock just like that.
Do you have any more video?
Very good observation.
This may actually be one that's called Atelocrinus. We have something in Hawaii called Atelocrinus conifer, and what I'm looking at is those bare filament ends on the arms on it, which is one of the characters, and plus the very long Seirios that it's propped up on is another character, I think.
Hey, whenever you're happy, video.
Let's
hold it
this
as long as
just another second.
Yep.
All right, coming out.
Hey, I got a scoot.
Oh, we're halfway to the end of the move.
Yeah, if you look at high pack, you can see how far away we are from the ship.
Yep.
Seems to line up with the current down here. Makes sense.
Okay with another move, 180?
Sounds good to me.
Yeah, I'm happy with that.
Okay, I'll let the... We still got 5 more meters or something.
Okay.
So, Diva, what is that? No, the thing that's on the rock. It's still there. It's waving.
On the left end?
In the center.
Yeah, I see.
Above the...
Yeah, I can't see right now. Either a crinoid or a brazingid. One of the two.
Coming in, video.
Caught in a very strong current.
Crinoid. Comatulid crinoid, I think.
It looks like it's in for the fight of its life.
I know, a lot of these animals do. And that may be why there's so little living up here, apart from some quite large and strong sponges.
Mind if I come in some more?
Go ahead.
Bridge nav. Yep,
You
we'd
know,
like
when
to
we
do another
were
move, please.
doing
30 meters,
our dive
bearing
on
180,
events, we talked a lot about
0.2
life
knots.
at the extremes, you know, at the
Right.
extreme of
When you're
temperature
ready.
and at the extremes of chemicals. And I feel like we're sort of looking at that same
Thank
kind
you,
of thing
Bridge.
now, that these animals have to live in an area where the currents are strong enough that they can get food, but they also have to make sure they're not in an area where they will get pulled off by the current. And now I'll go back to my regularly scheduled geology programming. Look at the rocks. We know what happens when I wax poetic about biology. I wonder what's up in the distance behind that sponge.
I think a slightly dead sponge.
A slightly dead sponge.
Yeah.
Not dying.
I mean, some of it looks
Slightly
white.
dead.
Okay, so a very dead sponge with some commensals.
Try to pull a zoom here, video.
Oh, Diva was right. Dead sponge with commensals.
Yep, it's got some squat lobsters, comatulid crinoids, perhaps some hydroids, and also it looks like a tiny live sponge up at the top right of it, as well as what looks like some barnacles.
All right, let's come out. Gotta move back a little bit.
Forward.
I just feel like all of a sudden we're really booking it. That's the current.
It's interesting. Now we're sort of in this flatter area now, with much less boulders,
and probably that
they've been
manganese crust. I heard that, navigator.
Come down a little bit, give you a little more to play with here.
Looks like we went off the slope a little bit.
Yeah.
Take us back west a bit more on the next move.
Another whip coral here, likely icidid or bamboo coral, perhaps from the genus Lepidicis.
Am I in video?
I might be tugging now.
And this one's got a couple chirostylid squat lobsters living on it, I think.
Actually, if the high point is the direction that we think it is, we need to go more west.
Yeah, we definitely need to go more west. We just came off the slope.
Okay.
Off the top.
So, actually, to get you back up there, I might go pretty drastic then, right? Like 230 or something?
Yeah, we do seem to be kind of on the edge of it. Yeah. I'll turn and face that way once.
We're just coming to the end of the...
Can you hear me out there, Diva?
Yes, I can, Scott.
This bamboo coral we're looking at looks a lot like one we collected from the Hawaiian cruise last year when we were in the Johnson Atoll. Down near the Johnson Atoll unit. And one of the things that's interesting is it also had a squat lobster. And so we took a collection with the squat lobster at the tip. There were some biologists who were interested in the squat lobster. And the squat lobster actually came off of the branch as we were collecting it and swam down to the bottom. But the pilot managed to maneuver the branch back down to the squat lobster and put it behind the squat lobster. The lobster grabbed onto the coral again, and then we collected them both, which really shows you that the lobster is intimately associated with that coral. It would have rather been on that coral piece than on the bottom.
And would you believe, Scott, that one of the pilots has already told me that story for the cruise?
It was a great story.
He actually went on to say that it didn't get onto the coral when the coral was in front of the lobster. It only backed onto the coral. It only got onto the coral when the coral was placed behind it.
They tell you who suggested to put the coral behind it.
Yes, yes, they did.
Come on in, video.
Partial.
So here's another small icidid.
Do you have any more coming in?
Full.
I'm turning to that 230.
Yeah, copy. Alright, come on up, video. I gotta push.